Good morning. This lecture video is going to go over the steps of DNA replication, or how DNA makes a copy of itself. Think about your skin cells. Roughly about every 30 days, the layer of skin that you're looking at right now, all of those cells die and they get sloughed off into the environment and they need to be replaced. We want all new cells that are being made to have all of the instructions necessary to making proteins, which is our DNA. So the cell has to figure out a way how to make a copy of DNA. And this is the process that the cell uses. It's actually really simple and pretty slick. First of all, just to summarize this, the stage of the cell cycle, so G1, the S phase, G2, and then the stages of mitosis, that's the cell life cycle. The S phase is going to be where DNA makes a copy of itself. S stands for synthesis, so we are going to be making, synthesis means to make, another molecule of DNA. And we want it to be exact, right? We want to have the exact instructions that make our cells specific to get passed on to the next cells. Basically, what's going to happen is that the two strands of DNA are going to separate. Then two new strands are going to insert themselves in between the originals, and then a proofreading process occurs, and we will then have two DNA molecules, each with two strands, each completely identical, and they'll wind themselves back up. One will stay in one cell and one will go into the new cell that's being reproduced. There are basically 4 steps to this, and I'm going to go over the 4 steps on the slideshow quick, and then I'm going to walk you through it on paper. There are 2 enzymes that are needed for DNA replication. The first is an enzyme called DNA helicase. DNA helicase is going to be the first enzyme that's going to be used, and its job is going to be to unzip and unwind the DNA strands, so that we're basically kind of going to make the DNA linear and spread them apart, spread the 2 strands apart. The second enzyme is DNA polymerase, and its job is to insert nitrogen bases into a new strand of DNA that will match the original strands. So 2 new strands are going to enter in between, and they're going to match up based on base pair rules, and the enzyme DNA polymerase is responsible for doing that. It's also responsible for proofreading this, to making sure that there are no errors so that the 2 DNA molecules that end up being formed are genetically identical. There are basically 3 steps to this process. Initiation, which is when the DNA helix unzips and unwinds the DNA. Elongation, which is when the 2 new strands come in between the original ones and then join by base pair rules. This is also where proofreading occurs. And then termination, when the 2 new DNA molecules will rewind themselves back up into that double helix shape. So I will show you what this looks like on paper. So we're going to start with the DNA strand. Remember, we got 2 strands of DNA, double-stranded. The backbone, the black, would be the sugar, the ribose, and the phosphate group. And then we have these nitrogen bases that are holding the 2 strands together. So A's join with T's, C's with G's, just like in the last lecture video. So the first thing that has to happen in this process, and I'm going to be oversimplifying this a little bit, but the 2 strands of DNA need to unzip and unwind. And this is done by a chemical or a molecule that's called DNA helicase. Basically, DNA helicase is going to come in here and it's going to break these 2 things apart so that we kind of get 2 linear strands here. All right, so if we follow this strand all the way down, okay, here's the A, C, keep going, T, C, follow the strand, G, T. All right, and then this strand over here, which would be this strand right here, okay, so this would be T, G, A, G, C, A. All right? So this strand here followed this strand, and this strand here followed that strand. Okay? So the DNA helicase comes in and it unzips and it unwinds the DNA. It separates this. Now, it doesn't actually do the entire strand of DNA. It would take way too long. So it's actually doing this in different sections all along the DNA at the same time. And the point at which it starts to unzip and unwind itself, we call that a replication fork. We don't need to get into all of the super duper process of that. Just know that the DNA doesn't completely do this all in one swoop. It's doing it all over the place in different parts of the DNA. So now that we have the DNA or sections of the DNA unzipped and unwound, the next step is that we're going to basically insert 2 new strands of DNA in between the old or the original strands. Okay, so here's the original strand. Here is an original strand. So now a chemical, an enzyme called DNA polymerase, is going to come in. It's going to insert 2 new strands right in between the original ones. And it's going to throw in the base pair that's needed to match up. Okay, so since this is an A, this has to be a T. Since this is a C, it has to be a G. So it's going to come in here and it's going to insert the strands and the matching nitrogen base. Okay, because these are floating all over, so it knows where it needs to throw it in. And now you can see we have 2 molecules of DNA. Okay, here's 1 molecule of DNA. Circle it kind of. Okay, here's the other molecule of DNA. And if you look, they are identical. Adenine, thymine, adenine, thymine, cytosine, guanine, cytosine, guanine. Okay, T-A, T-A. Each of the new molecules has 1 of the original strands from this, what we started with, and it has 1 new strand. Now, last thing that has to occur. This process is so important. We need to make sure that the letters, that there's been no mistake in the letters. Okay? So the DNA polymerase, it does a lot of different things, but the last thing that it does is it kind of goes through here and it proofreads. All right, if for some reason a T was put right here, it would be like, wrong, I need to go back, I need to fix that, and it'll put in the proper nitrogen base. So it will go through and it'll proofread the 2 new strands of DNA, and then when it's done, it's basically going to zip itself back up into that helix shape. And now we will end up with 2 new molecules, genetically identical molecules of DNA.